Hello, in this video we would be learning about CPA cocktail formulation using Taguchi method of experimental design. CPA cocktails have reduced toxicity as compared to single component CPAs such as dimethyl sulfoxide, ethylene glycol, propylene glycol, and methanol. Developing a CPA cocktail can be time consuming if done by optimizing single parameters sequentially. Traditional methods with the design of CPA cocktails involve the study of one parameter at a time, which require extensive combination of experiments that are time and labor intensive. An approach to simplifying this is the Taguchi method of experimental design. The Taguchi method allows a maximum number of parameters to be estimated in an unbiased fashion with a minimum number of experiments. Let us use a research study as an example and understand the problem at hand in some more detail. We would be looking at how researchers have used the Taguchi method to develop CPA cocktail solutions for the cryopreservation of mosquito larvae. The first step in developing CPA cocktails involves identifying the individual CPA solutions of interest. In this study, the CPA solutions of interest are dimethyl sulfoxide, ethylene glycol, and methanol. The next step is identifying the concentration at which these solutions can be added. Adding too much, as you can see, can be too toxic and lead to complete lethal dose. Adding too little of CPA can result in complete survival but lead to unsuccessful preservation. One way to optimize the survival versus toxicity is identifying the median lethal dose, which results in 50% survival. This is the LD50 value. Here, two different strains of mosquito larvae are considered, where the larvae were exposed to CPA for 1 hour and survival was quantified. Based on the survival data, as you can see here, LD50 values were identified. Here, we are focusing on the LD50 values and CPA cocktail design for Anopheles gambiae. Based on the LD50 values identified, we see that methanol is the least toxic while dimethyl sulfoxide is the most toxic. Now, we need to identify the concentrations at which these solutions can be added. The concentrations selected for each CPA are based upon the dose-response toxicity of single-component systems that we've seen before, where the least toxic CPA, that is methanol, is added at a relatively higher concentration, and the most toxic CPA, that is DMSO, is added at a relatively low concentration. Also, the concentrations are selected in such a manner that we ensure that the combined concentration is near the LD50 value of methanol on the higher side, while the LD50 value of DMSO on the lower side. In Taguchi methods, the parameters or CPA that are to be studied are termed as factors, as you can see here, and the concentrations are termed as levels. Here, in our case, we have 3 factors which are each studied at 3 levels. Based on this, the number of experiments can be defined as level minus 1 into the number of factors plus 1. This gives us the minimum number of experiments to be performed for this optimization is 7, and this is the notation that is used for the Taguchi experimental design. Based on the number of experiments required, we can refer to standard orthogonal arrays available. And based on our requirements, as you can see here, we need minimum number of factors are 3, and we need 3 levels for each of these factors. And also, the minimum number of experiments that are required are 7. So this gives us an option of L9, where the number of experiments to be performed are 9. We have to keep in mind that we can never use an array that has fewer rows or experiments compared to the number of experiments that we calculated using the formula. The number of factors of a given level in the table is a maximum. So you can have 3 factors for L9, but you cannot have 5 factors. And you can put a factor with fewer columns into a column that has more levels, but not fewer. So here, you have to have the maximum number of levels as 3 for each of these factors, and they cannot go beyond that. So now, based on the standard array that we have selected, here we see how the L9 array is organized and the order in which these levels are added for each factor. Since we have 3 factors, we will make use of only the first 3 columns. And based on this array, we will construct the array for the factors and levels that we have identified. So here we see an L9 orthogonal array constructed for the CPAs that are selected: dimethyl sulfoxide, methanol, ethylene glycol, and each at the 3 concentrations that we have chosen. This results in 9 unique combinations, which has a varying total molarity. Now, the same experiments that were done on individual CPA solutions are repeated for CPA cocktails for survival analysis. So the larvae were again exposed 1 hour to the each CPA cocktail, and then the survival was quantified. As you can see, the LD50 was found to be 3 molar, which is indeed higher than the LD50 values of individual CPAs. You can also see that 2 optimum CPAs were identified, which gave optimum survival. So this completes the Taguchi method of design for the CPA cocktails for use of cryopreservation in mosquito larvae. The Taguchi method has also been used for biostabilization of blood and developing CPA cocktails for zebrafish larvae. Thank you.